We're now going to go over an important section in pharmacology and discuss the therapies used to treat bacterial, viral, fungal, and parasitic infections. Guys and girls, this is a very high yield section, both for step 1 and beyond. These are life-saving drugs that you'll be using no matter what field of medicine you choose to go into. There are 5 things you will need to know about each class of medication: the drug names, the mechanisms of action, the clinical use, the toxicity, and the resistance pattern. This wonderful slide from First Aid gives us an overview of how we can treat bacterial infections. The key principle of antibacterial therapy is that the drugs we use only target cellular mechanisms found in the bacteria. If the drug is not specific for the bacterial cell, that is when we see adverse or toxic effects of the medication. There's going to be 6 cellular processes that are targeted by our antibacterial drugs. We can target cell wall, folic acid, DNA, protein, and mRNA synthesis, or we can damage the DNA, as seen here. We will start a discussion with the drugs that interfere with the bacterial cell wall. Before we start talking about penicillin G and V, let's think back to some basic bacteriology and talk about the cell wall. If we remember, peptidoglycan is the main component of the bacterial cell wall. If we remember, peptidoglycan is synthesized and then cross-linked in the bacteria. The medications that we have to treat bacteria that act on the cell wall can either mess with the synthesis or the cross-linking of the cell wall. The beta-lactam antibiotics are a big class of antibiotics that interfere with the peptidoglycan cross-linking. They all share a common chemical structure that includes a 4-membered beta-lactam ring. The 4 beta-lactam drug classes that we will go over are penicillins, cephalosporins, the monobactams, and the carbapenems. Penicillin G and penicillin V are the prototypical antibiotic that was discovered by Alexander Fleming in 1928. There are numerous derivatives of penicillin drugs, and all of their names end with illin. So if the drug ends with illin, you'll be chillin' because you know it's a penicillin. Real quick, let's look at this bacterial cell for a second. Do you know if this is a gram-negative or a gram-positive bacterial cell? And how would you know? Well, we know this is a gram-negative bacteria because of the presence of a periplasm. Gram-positive cells do not have a periplasm. Do you remember what the function is? Well, the periplasm is the space that is found in between the 2 membranes of the gram-negative bacteria and hold numerous different enzymes. Remember, gram-positive bacteria only have 1 membranous layer and therefore doesn't have a periplasm. As we said earlier, peptidoglycan is the main substance that makes up the bacterial cell wall. It is formed by alternating subunits of NAM and NAG. This stands for N-acetylmuramic acid and N-acetylglucosamine. The NAM subunit is conjugated to a polypeptide sublinker that I'm circling here. These peptides join together to cross-link the adjacent peptidoglycan strands. With this basic bacteriology in mind, do you remember what enzyme is responsible for cross-linking these polypeptide linkers? That is transpeptidase. Very good. With this basic in mind, do you remember the mechanism of action of the beta-lactams? Remember, all the beta-lactam drugs have the same mechanism of action, and these drugs bind and inhibit the transpeptidase enzyme. So without the transpeptidase enzyme, the adjacent peptidoglycan molecules are not able to cross-link, and therefore the cell wall is not able to form properly and the bacteria cannot survive. The original penicillins are penicillin G and V. Penicillin G is given IV, and penicillin V is given orally and can be vomited. An important point to remember about these 2 penicillin drugs is that they're beta-lactamase sensitive. To understand why these penicillins are sensitive to beta-lactamases, let's take a quick look at the chemical structure of penicillin, as seen here. Can you point to the screen where the beta-lactam ring is? Very good, it's right here, this 4-membered ring. The beta-lactam ring is found in all of the beta-lactam drugs, and it is the part of the molecule that is susceptible to cleavage by beta-lactamases. Bacteria express beta-lactamases, which destroy the beta-lactam ring and make the drug inactive, as shown here. The beta-lactamases act at this part of the beta-lactam ring, in between the carbonyl group and the nitrogen group. In the rest of the slides, we're going to use this lightning bolt here to illustrate the activity of beta-lactamases. An extremely important and highly tested topic about penicillins is knowing which ones are beta-lactamase sensitive and which ones are not. We will be discussing which ones they are and why in the upcoming slides. Penicillin G and V are what is called narrow-spectrum antibiotics. This means that the drugs only work on a narrow or small number of bacteria. I'll be representing narrow-spectrum drugs in the rest of the slides with this skier holding a sniper rifle. The sniper typically can only kill a few bacteria at a time, and that's why we'll be representing it with this guy here. Penicillin G and V are only effective against gram-positive organisms, such as Strep pneumo, Strep pyogenes, and Actinomyces. There are a few exceptions to this rule, including its activity against Neisseria meningitidis and Treponema pallidum. Thinking back to Treponema pallidum, do you remember what kind of microbe it is? Treponema is a spirochete, which I have represented here by this corkscrew, and it causes what disease, do you remember? It causes syphilis. Very good. Although these drugs are effective against Neisseria and some treponemas, as a general rule, these penicillins are most effective against gram-positive organisms. Common adverse effects of penicillins are classically hypersensitivity reactions, including rash, anaphylaxis, and hemolytic anemia. If we think back to immunology, what type of hypersensitivity reaction would cause rash and anaphylaxis? That would be a type 1 hypersensitivity reaction. Very good. Do you remember what immunoglobulin is responsible for mediating this type of hypersensitivity reaction? IgE. Very good. And what cell is activated by IgE? That would be the mast cell. Very good. And in response to activation by IgE, what molecule is released by the mast cell? That would be histamine. Very good. Another adverse reaction, like I said, is hemolytic anemia. This has a different mechanism of action, and in fact, this is what's called a type 2 hypersensitivity reaction. The way this happens is the penicillin drug binds to the red blood cell. So here we have a red blood cell. Imagine the penicillin drug binding to the membrane, causing a conformational change in the red blood cell membrane. As a result, immunoglobulins recognize this conformational change as foreign, bind, and then when the red blood cell goes through the spleen, it becomes hemolyzed and causes a hemolytic anemia. Now let's talk about the immunopenicillins, amoxicillin and ampicillin. One of the most important facts about these drugs is that they are beta-lactamase sensitive, similar to penicillin G and penicillin V. An important point to know also is that clever scientists and physicians came up with beta-lactamase inhibitors that protect our precious penicillins from beta-lactamases. Here we can see clavulanic acid acts to protect our penicillins by binding and inhibiting the beta-lactamase enzyme. Ampicillin is commonly given with clavulanic acid, and when it's given that way, the drug is beta-lactamase resistant. One of the most important parts about immunopenicillins is that they are broad-spectrum penicillins. I'll be representing broad-spectrum drugs by these two army guys carrying automatic rifles in comparison to the sniper rifle that we saw on the previous slide. But what does it mean when an antibiotic is broad-spectrum? Well, put simply, it means that the drug has effective against more types of bacteria. And why do we think that is? Well, if we remember back to the penicillin chemical structure shown here, the aminopenicillins differ in the R group portion of the molecule. We can see by the addition of this amino group, the ampicillin and amoxicillin have a better ability to cross into bacterial cell walls and bind to the transpeptidase. We can think of this amino group as a slippery coat that enables the drug to slip and slide through the cell wall and bind to that transpeptidase. Do you remember how penicillin G and V in general only covered gram-positive organisms? Well, thanks to the amino group here on the aminopenicillins, these drugs have a broader spectrum of coverage and can be used against gram-negative bacteria. They are amped up penicillins. A mnemonic to help remember the bacteria that are susceptible to these drugs is the mnemonic HELPS. It is used to remember that this drug is effective against Haemophilus influenza, Helicobacter pylori, E. coli, Listeria, Proteus, Salmonella, and Shigella. Do you remember the most common infection caused by E. coli in sexually active women? Yeah, that would be a UTI. Do you remember what life-threatening infection that H. influenza causes in children? Good, epiglottitis, very good. What kind of disease does H. pylori cause? Causes peptic ulcers and contribute to the formation of gastric malignancies. Very good. The toxicities associated with amoxicillin and ampicillin include rash, anaphylaxis, and pseudomembranous colitis, particularly for ampicillin. The mechanism of pseudomembranous colitis is that the antibiotic kills some of our normal gut flora, which allows for, do you remember which bacteria? Clostridium difficile, to grow and cause a colitis in the large intestine. Do you remember what major symptom you see in patients with C. diff colitis? Good, watery diarrhea, yes. The board examiners tell you someone has been recently treated with antibiotics, and then they are having numerous watery stools, you should be immediately thinking about C. diff colitis. Now let's talk about the next class of penicillins, called the penicillinase-resistant penicillins. Basically, this means that these drugs are resistant to the beta-lactamases. The drugs in this class include dicloxacillin, nafcillin, and oxacillin, and methicillin, although it is not listed here because it's not commonly used anymore. In comparison to the beta-lactamase-sensitive penicillins, such as the ones we just talked about these drugs are beta-lactamase resistant. Let's think back to the mechanism of the beta-lactamase. Remember, the beta-lactamase is an enzyme that cleaves the beta-lactam ring here, between the carbonyl group and the nitrogen group. So what would happen if the beta-lactamase enzyme is unable to reach this part of the molecule? What if there is a big, bulky chemical group protecting it from destruction? Wouldn't that be cool? Well, that's exactly what these drugs have. They have this R group that's big and bulky that protects this part of the molecule, the beta-lactam ring structure, from the enzyme. So you can imagine the beta-lactamases here not being able to reach this part of the molecule and therefore not being able to cleave the beta-lactam ring. However, when there is an upside, there's always a downside. Since these molecules have this bulky R group, they are less able to cross some cell walls. In fact, as a result of this, these drugs have a narrow spectrum of activity, meaning that these drugs are only effective against a few bacteria. See our sniper guy again. In practice, these drugs are used to treat Staph aureus, the infamous gram-positive cocci that is full of beta-lactamases. So remember, we use NAF, or nafcillin, for staph. Unfortunately, some Staph aureus bacteria become resistant to these drugs. Do you remember which infamous bacteria this would be? Correct, this is MRSA, or methicillin-resistant Staph aureus. MRSA has this annoying ability to mutate the genes that encode for its transpeptidase enzyme and causing an altered protein target. By doing so, the penicillin drugs are unable to bind and therefore are ineffective against MRSA. Like penicillin, the adverse reactions to these drugs include a type 1 hypersensitivity reaction, including rash and anaphylaxis. A unique reaction caused by methicillin is interstitial nephritis, which is the reason why the drug is no longer used. Now we're on to the last section of penicillins. Pat yourself on the back, guys and girls. We are almost done with one of the most important classes of antibiotics. The final and one of the most important classes of penicillins is the Antipseudomonal penicillins, piperacillin and ticarcillin. These drugs are sensitive to beta-lactamases, but are often given with a beta-lactamase inhibitor, tazobactam. These antibiotics have a broad spectrum and are bactericidal. And remember, notice our army buddies again here. They have activity against Pseudomonas and many gram-negative bacteria, which, if we remember correctly, are not covered by penicillin G or V. For any of you that has spent some time in the hospital, Zosyn, or piperacillin-tazobactam combo, is often frequently used. So in review of all the penicillins we have covered, we have penicillin G and V, we have the aminopenicillins, including ampicillin and amoxicillin, we have the penicillinase-resistant penicillins, including nafcillin and oxacillin, and then we have the antipseudomonal penicillins, ticarcillin and piperacillin. These are the four groups of penicillins that each have slightly different characteristics that make them a very powerful and useful class of drugs. When reviewing the penicillins, I want you to be comparing and contrasting them. I want you to know, number 1, are they narrow or broad spectrum? If so, why? Number 2, are they beta-lactamase susceptible or resistant? Why or why not? Number 3, are they given with a beta-lactamase inhibitor? If so, which one? And 4, what are the important adverse reactions caused by these drugs? How are they similar and different? If you can do all of this, you'll be in great shape for your USMLE Step 1. Now let's talk a little more about the beta-lactamase inhibitors. Remember, beta-lactamases are enzymes produced by bacteria that provide resistance to the beta-lactam antibiotics, like the penicillins for example. The enzymes cleave the beta-lactam ring structure here. Beta-lactamase inhibitors are drugs that we can give along with the penicillins that inhibit the activity of these beta-lactamase enzymes and protect the antibiotic. There are 4 important beta-lactamase inhibitors. Clavulanic acid, which is combined with what drug? The aminopenicillins. Very good. Ampicillin in particular. Then there's sulbactam, which is combined with what? Amoxicillin. Very good. And then there's tazobactam, which is combined with which penicillin? The Antipseudomonals, piperacillin, and ticarcillin. So, these drugs are a cast. Alright, now for a flash quiz. Which penicillin can lead to interstitial nephritis as an adverse effect? Very good, that is methicillin, one of the penicillinase-resistant penicillin drugs. In fact, interstitial nephritis as an adverse effect is the reason why we no longer use methicillin in the clinic.